بسم الله الرحمن الرحيم. This is the lecture in the immunology, second lecture. The references is medical microbiology by Jowett and immunology by Roy, immunology by Cooper. The objective of this lecture is to define antigen, types of the antigen, characteristics, and factors that determine antigen effectiveness. The antigen can be defined as any molecule that can be specifically recognized by the adaptive immune system, and this is the antigen. The epitope is a restricted part of the antigen, which is either short sequence of the sugar amino acid that bind with antibodies. This is the abbreviation of the antibodies. So this is the epitope. This is the whole is the antigen, and this is a small parts or knobs is epitope. Hapten is a small molecule had a low molecular weight that cannot initiate the immune response unless it's coupled with a large carrier molecule. So this is the hapten that not stimulate the immune system unless it's coupled with a large carrier molecules. So this hapten attached to this carrier molecules, which is called hapten carrier conjugate, and this leads to stimulate the immune system. Now we have the what's called term major histocompatibility complex, MHC. What's the MHC? Each mammalian species possesses a tightly linked cluster of genes, which is called major histocompatibility complex, M for the major, H for the histocompatibility, and C for the complex. And their products play a major role in cellular recognition in determining the transplanted tissue is accepted or rejected. So the product of these genes were expressed as antigens on the cell surface of the cell. It's also called the human leukocyte antigen, H for the human, L for the leukocyte, and A for the antigen. The MHC is consisted of three classes: class 1, class 2, and class 3. The superantigen, what's superantigen? Is antigen like staphylococcal enterotoxin that binds T cell receptor variable beta region to alpha chain of a class 2 major histocompatibility complex on the antigen presenting cell. This leads to stimulation of multiple T cells. So the normal antigen, which is sit in this space between the T cell receptor and the major histocompatibility complex class 2. While the superantigen is bind from the outside, it's bind to the beta chain of the T cell receptor, which is the variable region of the beta chain of the T cell receptor, and the alpha chain of the class 2 MHC complex. So this superantigen leads to stimulate large number of T cells with no benefit, because it is not lead to stimulate the T cell. It's only bind to its from the outside, that the T cell did not do its main function. So this leads to stimulate multiple T cells. And the best example of this is the staphylococcal enterotoxin, which is the bacterial secrete toxin that leads to gastroenteritis. Factors determining the antigenicity. Degree of foreignness. The more relative of the two individuals, the more similar of the antigens. So the transplantation between two identical twins, so there is no stimulation of the immune system because there is no foreignness between them, because they are identical twins, while two individuals not relate to each other. So there is high degree of foreignness that lead to stimulate the immune system when transplantation of the organs occurs. Second factor, molecular size. The best molecular size of the antigen is 100,000 Dalton. If it is smaller than this, it's called hapten. If it is too large, so it's not lead to proper stimulation of the immune system. Third, the chemical structure of the antigen. They found that proteins is best stimulate to immune system. Second, the polysaccharide. Third, lipid, and the last one, the nucleic acid. So the vaccine, they choose the protein from the pathogen to use as a vaccine because protein is best stimulation to the immune system. The another factor, susceptibility to antigen processing, which is the ability of the macrophage when it's phagocytosed the antigen, so it is processed, it's into small parts, so more processing of the antigen is best to stimulate the immune system, while when the macrophage phagocytosed the antigen and did not process it and fragmented into small pieces, so this lead to not stimulate the immune system. Genotype. We found that some families have the susceptibility to some disease because of the genetic factor, while the other families resist or not develop same disease because they did not have the genetic susceptibility to develop this disease. So the genetic factor is important in determining the antigenicity. The dose of the immunogene. It's common sense that the high dose of the immunogene so it's lead to stimulate the immune system, while the small dose of the immunogene it's lead to less stimulation of the immune system. Route of administration of the antigen or immunogene. We found that the intravenous administration of the antigen is lead to faster stimulation of the immune system than the subcutaneous administration of the antigen. And this is found in the drug administration. When we give the drug intravenously, so it is lead to allergy and anaphylaxis while faster than when give it subcutaneously. Adjuvant. Some antigen used other substance coupled with it to increase its antigenicity, and this is substance mixed with the antigen to enhance the immunogenicity. For example, aluminum, potassium sulfate that usually used with the vaccine to enhance the immunogenicity and increase the susceptibility of the immune system and more stimulation to the immune system. And thank you.